A recent study on bats has shed light on how the human brain is intrinsically wired for collective behavior. This study delved into the intricate ways in which bats navigate and interact with each other, revealing that certain regions of the brain are activated during collective behavior. These findings provide insights into the evolution of social behavior and the neurological processes that underlie it. Bats' ability to navigate through space may be linked to their proficiency in social environments. The same neurons responsible for spatial navigation may play an important role in guiding bats through their social interactions. In a recent publication in the journal Nature, it was discovered that the neurons which aid bats in their spatial navigation also assist them in navigating complex social environments as a collective. The hippocampus is a brain structure that is thought to assist in navigation for many mammals, including humans and bats. It creates a mental map of familiar surroundings, allowing individuals to understand their environment. As one moves about their neighborhood or travels to work, their hippocampus produces place neurons that signal their location. The researchers at the University of California, Berkeley conducted a new study where they employed wireless neural recording and imaging tools to eavesdrop on the hippocampal brain activity of Egyptian fruit bats. These bats flew freely in a spacious flight room, often navigating through dense social groups while movement trackers documented their actions. Upon examining the bat's social environment, the researchers were taken aback by the amount of information that was encoded by the bat's place neurons. These neurons did not just represent the animal's location, but also contained information about the presence or absence of another bat at the location the bat was flying towards. Furthermore, when another bat was present, the activity of these neurons indicated the identity of the bat they were flying towards. According to Michael Yartsar, an associate professor of bioengineering and neuroscience at UC Berkeley and senior author of the study, this is among the initial papers to demonstrate the representation of identity in a non-primate brain. Interestingly, the researchers discovered this phenomenon in the supposed center of the brain's GPS. They found that this area still functions as a GPS, but with an added sensitivity to the social dynamics present in the environment. According to Angelo Forlì, the principal author of the study and a postdoctoral member of Yartsev's Neurobat lab, humans and bats, despite not being as visually striking as schools of fish or flocks of birds, still exhibit collective behavior due to their high level of socialization. According to Forlì, social creatures, much like humans, will synchronize their movements within a given space to accomplish a variety of objectives. These objectives can range from simply visiting others to traveling together in a coordinated manner, such as in classical collective behaviors or during a game of soccer. Alternatively, these objectives can manifest themselves in other forms of collaboration or even opposition. At first, Forlì was skeptical about the effectiveness of allowing groups of bats to interact and fly freely in order to gain insight into the neural basis of collective behavior. The complexity of the experiment caused him to doubt whether significant results could be obtained. His main concern was that the social interactions and movements of the bats could be too random to reveal any strong correlations between their behavior and neural activity. He was taken aback when he noticed that the bats had established a few specific places to rest in the flight room on their own and were following similar routes when traveling between them. Additionally, the bats exhibited a clear inclination towards certain friend bats, with a tendency to fly towards them and often landing in close proximity or even on top of one another. According to Forlì, their findings revealed that when a few bats were placed in a single room, their actions were not random. Rather, they exhibited specific behavioral patterns that were consistent. These patterns included tendencies to spend time with particular individuals and to favor specific, unchanging locations. Forlì was able to distinguish the neural activity linked to diverse flight paths by utilizing exact behavioral patterns. Additionally, Forlì was able to ascertain how neural activity was affected by the presence of specific bats at the destination and the movements of other bats. Yartsev stated that by monitoring only a few neurons in this particular part of the brain, the actions of the bats within their social environment could be accurately determined. It would be possible to discern whether the bats were heading to an area devoid of other bats or if they were actively seeking out the presence of other bats, which Yartsev found to be an unexpected discovery. During the past few years, Yartsev and the NeuroBat Lab have implemented a range of wireless devices for neural recording and flight tracking technologies to reveal several unexpected insights about the brain. These discoveries include the synchronization of neural activity while bats socialize, the role of the frontal cortex in distinguishing self from others during vocal communication, the hippocampus's ability to map flight paths in addition to specific locations, and the potential for permanent spatial memory to be stored in the brain. The team's research on navigation and social behavior is combined in this new study, demonstrating the deep-seated connection between the two in the brain. These findings also bring to light the reason why damage to the hippocampus has been linked to the loss of both spatial and social memory in neurodegenerative diseases like Alzheimer's. Yartsev stated that our memories of specific events are a union of the milieu we are situated in and the experiences we have within that milieu, which involves our social interactions. It is unexpected that this connection has not been observed previously among groups of animals and at the level of individual neurons. However, it is also logical in light of the deficiencies that individuals with impairment to the hippocampus encounter. According to Yartsev, this study emphasizes a crucial aspect of neuroscience research. Many neuroscientists investigate the brain in artificial or simplified conditions that do not reflect the brain's natural behavior and functions which it has evolved to support. This study highlights the significance of using a natural approach to neuroscience research and demonstrates its effectiveness. According to Yartsev, the study of place neurons has been ongoing for 50 years, but the majority of this research has centered around single animals navigating an empty container. Yartsev's research indicated that there is significant potential for learning when neuroscience research is shifted towards observing natural forms of behavior. This study received funding from several sources, including the New York Stem Cell Foundation, NYSCFR-NI 40, the National Institute of Mental Health, Award 1 R01MH 2538701, the Air Force Office of Scientific Research, FA 955017104112, the Packard Fellowship 2017 66825, the National Institute of Neurological Disorders and Stroke R01 NS11842201, the Valley Foundation VS 2000 2034, the Office of Naval Research N00014211263, the Searle Scholars Program SSP 2016-1412, the Human Frontiers Fellowship LT 000302/2020, and the European Molecular Biology Organization 1022-2019. To support our channel's growth and ensure broader awareness, kindly hit the like and subscribe buttons. This will help us reach more individuals and disseminate valuable information. Thank you in advance.